Hi friends, so in this video we are going to talk about Mendelian genetics, different crosses like monohybrid cross, back cross, test cross and dihybrid cross which are used to check like the parents genotype if they are like uh, dominant or if they are like homozygous or heterozygous, like in case of the test cross and back cross to generate the pure lines as well, and the difference between monohybrid and dihybrid cross. So please make sure to like, share and subscribe my channel. So, first we will talk about the monohybrid cross. So, monohybrid cross is a cross between two individuals that differ only in one character. Like, suppose we are talking about the male and female, in which all the genes character are same, and what determines we are talking about only one character, which is like height, suppose, or the flower color, or some, or like suppose one flower has the red color and the other one is white, so we are talking about only one character. So when we are crossing this individual in which we are studying only one character is called monohybrid cross, and then character depends only on one gene. So here you can see we are talking about the color which is yellow or green. So in this case we have the dominant one. So in my previous video I have talked about what is dominant and what is recessive, what is homozygous and what is heterozygous. So just in brief, the dominant is something which in which one allele overrides, like mask the expression of the another one. So as you can see in this case, yellow, because it's represented by uppercase letter, it's the dominant one. The yellow color is expressed dominantly, and the small y, which is like a lowercase letter, this is the recessive color, which is green in color. So when they segregate, like the based on the law of segregation, the gametes that we get is capital Y or small y from here. And when these two cross, what we get is capital Y from here and small y from here. And this is called the heterozygous. Heterozygous means in which both the alleles, because one gene is represented by two alleles, right? When both the alleles are different. So here you can see both the alleles are capital Y. So this is called homozygous. But in this case, there is one capital allele and the another one is small allele. So this is called heterozygous. And here we can see that we have one capital Y, one small y, but the color that we get is the yellow one, which is from the capital one. So this means that the capital Y is dominant over small y. So this is called the first generation because this directly from the parents. And for the second generation, what we do is we take this F1 generation and cross it again with the F1 generation, like males and females. And when we put this in a box, in a square, so this is called the Punnett square, which is used to represent all these crosses, right? So when we put this in a Punnett square, what we get is 3 with the dominant one, like either homozygous or heterozygous. Like here you can see both homozygous, heterozygous, and this is homozygous recessive. So what we get is 3 with yellow color and 1 with the green color. So this is by the appearance, which means that by appearance, if you see, we have yellow, but the genotype they have, like they can be either homozygous or heterozygous, is different. So what we put here is phenotypic ratio. Phenotypic means which you can see like the appearance. So what we get is 3 yellow and 1 green. So the ratio is 3:1. But if we go for genotype, like homozygous dominant, homozygous recessive, or heterozygous, so we have one homozygous dominant, two heterozygous, and one homozygous recessive. So the genotypic ratio is 1:2:1. So according to Mendelian genetics, Mendelian inheritance, in monohybrid cross, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. So But like in my previous video, we have talked about the incomplete dominance, codominance, in which this ratio changes, right? The genotypic, like the phenotypic ratio as well, because in that case, the like in case of incomplete dominance, it's not completely dominant, and the phenotype that we get is the intermediate of both the parents. So based on the different conditions, this ratio changes for the monohybrid cross as well. So now what we are going to talk about is back cross. So as the name suggests, back cross means we are crossing it back with something. So what is that something is? So what we get is a F1 generation, right? Like we have talked about this intermediate, like the heterozygous one is the F1 generation. So what we do is we cross it with any of the parent. So if we cross with this F1 with either this dominant one or this recessive one, so when we do this, like crossing the F1 generation with its parents, it's called the back cross. And why it is done mainly to achieve the like the genetic identity or to generate pure lines. Because if you just like back cross again and again and again and again the F1 with the parents, what you get is like the chances of recombination are less, and what you get is mostly all the genes from the parents. So what we are generating is pure lines that are closely related to their parents. So backcross is done to generate the pure lines, like the genetically identical lines with the parents. So what you here I have showing you the example of the F1 cross with the dominant parent. It doesn't need to be specifically dominant parent, it can be the recessive parent as well. So in this case I am just showing you the dominant one. So here you can see we are taking this dominant parental generation. So in the beginning it's the same as the monohybrid cross, we get the F1 generation, and we from with this F1 generation we are crossing the capital Y, as you can see here the parent and F1 generation here. So in this case, as you can see, we did not get any of the green seeds, because if you cross them, in all of them you will get at least one dominant Y. And in this way we are making sure that every time you do this, we are going more closer to the parent, so we will get all the dominant seed, like which parents we want to go closer. So like if you do this with hair, maybe you can still get like the small y, so it changes. So as you can say, this is one of the cross in which you will not get the exact ratio of monohybrid cross like 1 is to 2 is to 1, but what we get is 1 is to 1 genotypic, because two of them are homozygous dominant and two of them are heterozygous. So the back cross is can be with either parents like the dominant one or the recessive one. So in this case, the back cross with the recessive parents is also called the test cross. Why? So the test cross is done to find out the genotype of any plant or if anything that you want to know. Like you see here, like with the phenotypic ratio, we can say that all of them are yellow seed, but we don't know if this yellow seed is because both the alleles are dominant, like the genotype is homozygous dominant or the heterozygous dominant. So because now we are doing this, and practically what we can say, it should be like this. But sometimes we just know the color, like it's a yellow in color, but we don't know what is the genotype, if it's the like the homozygous dominant or heterozygous. In that case, to identify whether it's homozygous dominant or heterozygous dominant, what we use is test cross. So what we do is, in the test cross, the homozygous or heterozygous like the allele which we don't know is crossed with the recessive genotype. For example, you can see here we have this flower, right, with the recessive genotype, like the small p, and what we are crossing it with the purple flower, which can be either both capital P like the homozygous dominant, or heterozygous like one capital P and the other small p, and in that case also the color of the flower will be purple. So we want to know this, so we cross them with the white flower which is homozygous recessive, and then based on the both are capital, or like one is capital and one is small, we get different ratios. Like if both are capital, what we get is all the purple flowers, but then if it's heterozygous, what we get is 1:1 ratio. So this test is used to know the genotype of whether it's homozygous dominant or it's heterozygous. And the condition is, in this cross, the 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 unknown genotype that we want to check is always crossed with the recessive one, like the it's always crossed with the recessive parent or something. So if you see, this is the F1 generation, suppose, and we are crossing it with the recessive parents. So in that sense it resembles the back cross, in which we are crossing the F1 generation instead of the dominant parent with the recessive parent. So test cross can be back cross when we are doing it with the back cross can be test cross when we are crossing the F1 generation with the recessive parent. So just to summarize, the difference between the back cross and test cross is, the test cross is the cross that occurs between a dominant phenotype, which can be homozygous or heterozygous, and a recessive phenotype, the recessive parents. But the back cross, in the back cross, it doesn't matter whether you are taking the recessive parents or dominant parent, it can be with one of the two parent. So test cross is like the, as we have told about, is the dominant phenotype. It can be from F1 generation or it can be from any other with its recessive phenotype. But back cross is always with the F1 hybrid with one of its parents. It should be test. All test crosses are back crosses because we are crossing with the recessive parent, right? But back cross with the F1 hybrid with the recessive can be considered as the test cross, but not always, because if we crosses with the dominant phenotype, it cannot be considered as test cross. So now we know what is monohybrid cross, how it can be used for the test cross, back cross, what is the ratio. So now what we are going to talk about is the dihybrid cross. So in case of dihybrid cross, what we talk about is like one character, or like or we are talking about like two different characters. Like in monohybrid cross, we are talking about only one character, suppose height or the flower color. But in dihybrid cross, what we are talking about, two characters. Like here you can see, first is the color, it can be yellow and green like the monohybrid cross, and then we are putting another character which is the round shape or the wrinkled shape. So we are talking both about the shape and the color. So in this case we are talking about the two characters, and the segregation of these two characters depends on both the characters. So here you can see the yellow color and the round shape is the dominant phenotype, while the green color and the wrinkled shape is the recessive phenotype. So when we do the cross and the F1 generation, that we get is, from here you get capital Y, capital R, and from here you get small y, small r. So what you get is this thing, in which it's heterozygous but the dominant one, right? So the phenotype that we get is yellow color. And when we cross this, the gametes from this F1 generation, right? So the gametes that we get, it involves the segregation from both the genes. So what you can get, this capital Y can be segregated with capital R, or it can also be segregated with small r, right? Same with this small y, it can be segregated with this capital R here, or it can be segregated with this small r here. The same here. So the segregation in which the it depends on the other trait as well is called the independent assortment, because they all can be independently segregated. Like this y will not always come with the capital R, it can also come with the small r. So when we cross this when we cross this F1 generation, what we get is different genotype, right? Which is completely like a mess to count the different genotype, because there are so many, like 16 in total. So what we calculate is mostly the phenotype based on the color and the shape. So here you can see what we get is 9 with the yellow color, like here, in which either both of the genes are present, the dominant alleles are present, right? So 9 with yellow color and the round shape, 3 with the green color and the round shape, and then 3 with the yellow color but wrinkles shape, because in this the r that is present, the which controls the shape, is always in recessive, and the one in which all the alleles like caps y and r is always recessive. So the ratio that we get is 9 is to 3 is to 3 is to 1. So this is the Mendelian ratio for dihybrid cross. But there are conditions in which this ratio changes. Like instead of getting this 9 is to 3 is to 3 is to 1, we get different ratio, that is called the non-Mendelian inheritance, that depends on the expression of other genes, and that is also affected by different genes, which I am going to talk in my next video. So I hope you like it, and if you have any doubts, please let me know in the comment section. Please like, share and subscribe my channel. Thank you.